It's Salwete. We are going to do this video lesson a little bit different. One difference is you will see my face. Hello. I hope that this is not a mistake and that this will be okay. We'll see how it goes. Second, I want to change up how I was discussing cell respiration. We were going to go in order of events, like the things that happen first in order to make all that ATP. So the order is glycolysis, link reaction, Krebs cycle, oxidative phosphorylation. After thinking about it, I decided I want to actually go backwards. So we're going to start with oxidative phosphorylation and build our way backwards. I think it'll be easier to understand, especially after students were saying the way that the information was presented was a little bit intimidating, and I totally get it. It's a lot of information. So we're going to change it up a little bit and go backwards. So let's just start very generally. Cell respiration. What is the point? Why do our cells respirate? Give yourselves a moment to think. Hopefully you're saying to yourself to make ATP, and that's correct. The reason why our cells respirate is to perform, is to make ATP in order for the cells to perform whatever functions they need to perform. So all cell biochemical pathways require energy. A lot of that energy is going to come from this molecule ATP. So we'll notice here that ATP is going to be produced in large quantities from this section of cell respiration, oxidative phosphorylation. It's going to make a big chunk of our ATP. And how does it do that? How do we get there? How do we get all that ATP? The first thing I want to talk about is the enzyme that is actually putting ATP together. And that enzyme is called ATP synthase. So it's just like the name, ATP synthase. You know that it is going to synthesize some ATP. And the way that it works is it takes an adenosine diphosphate molecule and an inorganic phosphate molecule, and it uses energy to push them together. And when it pushes them together, it then makes adenosine triphosphate. And that ATP can go elsewhere in the cell. So the way that this protein works is quite fascinating. It looks like a motor. It is one of the first proteins that I learned about where I was like, whoa, this is in our cells. Like, this is crazy. It looks like a tiny machine, but it's not a man-made machine. It's a cell-made machine. And that's crazy. It's so cool. I also want you to note that it is embedded within a membrane. So in year one, we learned about different functions of cell membrane proteins. You had to remember that word or the name Tracy as a mnemonic to remember the different functions. And the E in Tracy stood for enzymatic activity. So here is an example of an enzyme that exists within a membrane and also performs this catalytic activity. So I want you to watch this video. I'm not going to play it in this recording, but you can pause this recording and then click the link and watch the video, and you can see a little bit of how it works. I am going to play it just a little bit so I can talk about how cool it is in this recording. I'm going to skip. So here we can see it working. So what's going on here is you can see that the membrane is separating some molecules. There's a lot of these tiny molecules that are floating around, and there's a smaller amount of those molecules on the other side of the membrane. Hopefully you're thinking, oh, this looks like a gradient of some sort. We see a high concentration of one thing on one side and a lower concentration of that same thing on the other side. And that's exactly what's happening. We have something called a proton gradient, and that gradient potential is going to provide the energy necessary to allow this motor protein to do its job. So hopefully you can pause this video and get a chance to watch how this enzyme works. It's very cool. The details in this video are a bit much, and you don't need to know all of it. The main things that you need to know are that it's a motor protein embedded within the mitochondrial inner membrane, and it is where ATP is made and how ATP is made from adenosine diphosphate and inorganic phosphate. So those are the main things you need to know for ATP synthase. Okay, so how does ATP synthase work? I want you to note that this particular diagram is in the opposite orientation than the video. So in the video, the concentration gradient is higher at the bottom of the image, and the motor, where ATP is synthesized, is at the top. In this image, it's flipped, and that's totally fine because these are going to be in different orientations depending on where it exists in the membrane and how we are looking at the mitochondria. So Always orient yourself when you're looking at your images to make sure you are interpreting what you're looking at the way that it was intended to be interpreted. So here, our concentration gradient, the high concentration gradient is going to be at the top of the image, and ATP is going to be synthesized at the bottom of the image. So the way ATP synthase works is kind of like a water wheel. So think of how a water wheel works. There's a flow of water hitting a wheel, and as it hits the wheel is going to turn. And so we have energy coming from the water, and it's being pulled down by gravity, and that energy is going to be transferred to the wheel. So it's going in a circle. And that's really similar to ATP synthase. In this analogy, the water would be the hydrogen ions, and the water wheel would be ATP synthase. So as the hydrogen ion or the proton gradient increases, we are establishing this gradient. And when the gradient is established, our protons can move down the gradient through ATP synthase. And as it moves down the gradient, you see it pops out down here. As it moves down, it's providing energy for the protein to spin. And as it is spinning or rotating, that is also creating energy to produce ATP from ADP and P. Okay, so just imagine a water wheel. I have a GIF somewhere here that we can reference. Let me find it, or a picture. So here, it's very similar to something like this. So if you're imagining water moving and that water is causing a wheel to spin, that spinning wheel's energy can be transferred as like through the connections that are made in this particular machine. So it's very similar to how ATP synthase works. All right, let's go back. Okay, so now we know that ATP synthase is powered by a proton gradient, right? Protons are going to flow through ATP synthase to get it to turn. How is that proton gradient established? Where does it come from? It doesn't just happen out of nowhere. Those protons have to come from somewhere. So the protons are actually coming from a protein pump. So there's a protein pump that is adjacent to or very close to ATP synthase. And that proton pump is going to take ions from the matrix of the mitochondria, and it's going to pump it into the inner membrane space of the mitochondria. And when it does that, it can start to accumulate protons in the inner membrane space. In this particular diagram, we only see one protein, but there's actually more than one. So here's another diagram depicting the same thing, just with more detail. Here we have our ATP synthase. We see our hydrogen ions are flowing down the gradient, and as they flow down the gradient, it provides ATP synthase with the energy it needs to push adenosine diphosphate and inorganic phosphate together to form ATP. So here we can see that there are protons being pumped into the inner membrane space by multiple proteins. So, let me talk about this term chemiosmosis. So the way that ATP synthase is working is by a process called chemiosmosis. So chemiosmosis is just a fancy way for saying diffusion across a membrane with ions. So chemiosmosis is just ions that are able to move across a semi-permeable membrane through a particular protein. That's what that means. And so this chemiosmosis is able to happen by establishing a gradient of hydrogen ions. So what's the point of chemiosmosis? Just as a review, so the point of chemiosmosis is to activate ATP synthase. ATP synthase can only work by the process of chemiosmosis. How is chemiosmosis achieved? It's achieved by establishing a proton gradient with the help or with the electron transport chain. So we have these proteins here that are pumping these ions or pumping these protons into a particular space to create a gradient. Okay, that's the point. So the point of ATP synthase is to make ATP. ATP synthase is powered by chemiosmosis. The point of chemiosmosis is to power ATP synthase. How do we get chemiosmosis? We have to establish a protein or a proton gradient. How do we establish a proton gradient? By using the electron transport chain. So the next question is, how does the electron transport chain work? How does it pump these protons across the gradient, because we know that protein pumps require energy. So where are they getting this energy from? Hopefully you're thinking, oh, they must get them from electrons. That's why it's called the electron transport chain. So here is a series of steps, and we see steps 1, 2, 3, and 4, and those steps are corresponding to these numbers that are placed on the diagram. So the electrons are actually coming from our electron carriers. If you remember us talking about NADH and FADH2. So NAD plus and FAD/NADH and FADH2 are going to be our electron carriers. So earlier in cell respiration, these molecules were able to get reduced and hold on to some electrons and hydrogen ions. They're going to drop off those electrons and hydrogen ions at the electron transport chain. So here we can see, let's just read this first step. Electron carriers from earlier parts of respiration deliver electrons and hydrogens to the ETC. NADH is oxidized and the electron transport chain is reduced. Very cool, right? So we see NADH here. We see this arrow. NADH is getting converted into NAD+. So NADH is getting oxidized into NAD+ because it's losing electrons. Those electrons are going to the electron transport chain. And the hydrogen ions are going into the matrix of the mitochondria. As soon as this first protein of the ETC receives the electron, it now has energy to pump the hydrogen from the matrix to the inner membrane space. So here is the first part of the electron transport chain. That electron is then going to get transferred to the next set or the next protein in the ETC, and that is going to do the same thing. It's going to pump protons into the inner membrane space. We also have FADH2 getting oxidized to FAD. And when this happens, it's going to release its electrons. Those electrons will join the electron transport chain and also pump more hydrogen ions into the inner membrane space. And so our electrons are going to travel through these proteins until it gets to this last part. And it doesn't stop here. These electrons have to be received by a final acceptor of electrons. And that final acceptor of electrons is going to be oxygen. So oxygen is going to react with electrons and hydrogen ions to form water. And so this is where water comes from our cell respiration equation. And this is where oxygen is necessary in our cell respiration equation. So we need oxygen to be our final acceptor of electrons. If there is no final acceptor of electrons, that the final electron will get stuck in this protein. And if it gets stuck in this protein, it's not going to move out. So if it doesn't move away, then there's not going to be space for the next electron to come through, and if there's no opportunity for that, we can't pump our hydrogens anymore. So we need to continually be moving these electrons through the ETC so that we can create more space for the newer electrons or the charged electrons to come through. And so those electrons the final acceptor of electrons is oxygen, and when oxygen accepts these electrons and hydrogen, it's going to become water. All right. So that's it. That's all we're going to cover today. I hope it wasn't too much and I hope it was digestible. So let's just summarize everything. You should be able to answer these questions. And they were supposed to pop up one by one, but I set this up in a weird way, so it's not going to pop up. So we'll just answer these questions while seeing all of them here. So first question, what is the point of cell respiration? Hopefully you're thinking to make ATP. Question 2, which molecule is responsible for producing large amounts of ATP molecules? ATP synthase. Which process facilitates ATP synthase? Hopefully you're thinking chemiosmosis. Question 4, what needs to be established for chemiosmosis to work? A gradient, specifically a gradient of hydrogen ions or protons, because hydrogen ions are protons. How do those protons get there? Hopefully you're thinking they get there by the electron transport chain. The electron transport chain, how is it powered? It's powered by electrons. Those electrons come from our electron carriers NADH and FADH2. Question 7. Which molecule is the final acceptor of electrons, and what does this produce? Oxygen. Oxygen is the final acceptor of electrons, and this produces water. And where does this all take place? This all takes place in the mitochondria, specifically within the inner membrane of the mitochondria. The proton gradient, or the high concentration of protons, is going to be in the inner membrane space between the two mitochondrial membranes, and ATP is going to be produced in the matrix of the mitochondria. If you just need a diagram for orientation of like where we are, I'm going to show you, this picture. So here is our double membrane, outer membrane, inner membrane. The darker red is the inner membrane space, and the lighter orange salmon color is the matrix. So this dark red is where all of our protons are going to accumulate, and they're going to pass through the membrane through ATP synthase. ATP synthase is going to synthesize ATP, and that ATP will be made in the matrix of the mitochondria. And that's it. If you have questions, let me know. If you have feedback, let me know. Hopefully this was a little bit easier to take in, and then I'll in a separate video, I'll go over the Krebs cycle, the link reaction, and glycolysis. All right. Have a great day, everybody. Take care. I'll see you later.